Now we will move on to the third stage of cellular respiration, which is respiratory chain. So first of all, we will see that how we harvest energy by extracting electrons. So as we know that when an atom shifts its electron from one atom to another, it also transfers its potential energy. Likewise, when the electrons from food are shifted and they undergo the process of electron transport chain, the electrons are transported from electron carriers and they activate the pump. As they transfer the potential energy, they are successful in activating the pump, which then yields the ATP and which is the available energy form. So you can see here from the diagram that when the electron move through the series of electron carriers, they transfer their energy, which is then used for the formation of ATP. And finally, there is an electron acceptor, which we discussed earlier, which is oxygen. It accepts electron and then the formation of water also occurs. So here you can see the detail of electron transport chain. You can observe that the first of the proteins to receive electron is a complex membrane embedded enzyme called NADH dehydrogenase complex. Carrier called ubiquitin then passes the electron to a protein cytochrome complex called the BC1 complex. This complex, along with others in the chain, you can see here the other complex, they operate as a proton pump. They drive a proton across this mitochondrial membrane, and the electron is then carried by another carrier, which is called as a cytochrome C to the cytochrome oxidase complex. This is cytochrome oxidase complex. This complex uses 4 such electrons to reduce a molecule of oxygen, and each oxygen then combines with 2 hydrogen ions to form water molecule. This series of membrane associated electron carriers is collectively called the electron transport chain, and you can see that NADH contributes its electron to the first of the electron transport chain, the NADH dehydrogenase. FADH, which is also attached to the inner membrane of mitochondrial matrix, it also feeds its electron into the electron transport chain and later to ubiquitin, and it is the availability of plentiful electron acceptor of which is oxygen that makes oxidative respiration possible. So you can see here the electron pump which was formed here due to this electron transfer. There the proton concentration increases, and due to this they operate the pump. Due to the transfer or coming of the protons from this side to this side, this pump operates and it results in the formation of a ATP molecule. Now we will see how this pump operates individually. This is called oxidative phosphorylation, or the process of ATP formation is oxidative phosphorylation, or it is also called as chemiosmosis. So you can see here due to the electron transport chain, which includes proton pump, this is high concentration of hydrogen here, and when these protons move from this side of membrane to the inner mitochondrial side, this pump actually operates and it results in the conversion of ADP to ATP molecules. And this is showing the overview of the process again. Actually, we will see what happens during the whole process of cellular respiration. The pyruvate is formed during the process of glycolysis, and we know that pyruvate is formed in the cytoplasm, and then it is shifted to the mitochondria where the process of Krebs cycle occurs, and then it results in the formation of ATP and certain electron carriers, and these electron carriers are then shifted to this mitochondrial membrane, and then the process of electron transport chain occurs here, and electrons are harvested and carried through this whole transport system. And as a result of the transport of electron, here the number of proton increases, and due to the proton gradient, you will observe here that the ATP pump operates, and due to movement of proton from higher concentration to lower concentration, this pump is activated, and finally it results in the formation of ATP molecule, and as we have discussed earlier, the oxygen molecules combined with the hydrogen molecule to form water molecule. Now we will discuss the output of electron transport chain. Actually, one NADH molecule can activate 3 pumps to yield ATP molecule. So 3 ATP molecules are released from one NADH, and 2 pumps are activated by one FADH2 molecule. So 2 ATPs are formed from one FADH2. So as we had 10 NADH, it will result in the formation of 30 ATP molecules, and as we have 2 FADH2 molecules, it will result in the formation of 4 ATP. So net gain is 34 ATP molecules. Now we will discuss the overall ATP theoretical yield during the whole process of cellular respiration. We can see here that 2 ATP molecules are being formed during the process of glycolysis, and 6 ATP molecules are formed from these 2 NADH molecules during the conversion of pyruvate to acetyl coenzyme, and then 2 ATP molecules are formed during this process of Krebs cycle, and then 18 ATP molecules are generated from these 6 NADH molecules, and 4 ATP molecules are again yielded from 2 FADH2 molecules. Now we have to see that here 4 molecules of ATP are being formed from 2 NADH. The question arises, why? Why not 6? This is because of the fact because we have to transport these electrons from cytoplasm to the mitochondria during the process of electron transport chain. So 2 ATP molecules are utilized during the transfer of these 2 NADH molecules to the mitochondria. That is why the ATP theoretical yield is 36 instead of 38. So this is all about the process of respiratory chain in specific and cellular respiration in general.